Good morning, students. In the continuation of the MSc1, endogenous pathway is another topic. Endogenous means endo means inner. So here in the cell, these pathways will occur. So that's why it is termed it as another name. It is termed it as cytosolic pathway. Cytosol means in the cell. Normally, protein turnover is regulated. It is achieved by the particularly chaperones. Actually, the lifetime it is termed it as half-life. So here the half-life for the damaged proteins, malforming proteins have less. Maybe it is less than 10 minutes. If it is functionable, it is a good. Its lifetime is 2 days. It is if it is what is called damaged and denatured abnormal proteins have very less half time. How it is achieved? So here, if it is a protein, it is a or it is a damaged protein, or it is a abnormal protein, or it is a abnormal protein, or maybe it is generated by the viral activity in the cell. These proteins need to eliminate, need to damage. Here, not only the protein, total cell is goes to the damage. So how it is achieved? Here, ubiquitin, it is a protein, will attach to the protein. So now these complexes conjugate with the proteasome. Proteasome means it is a 20S, and also it contains 14 subunits in it is 14 subunits. It is a tubular hollow one. In this hollow path, in this lumen, these proteins, the peptide bond between the proteins is cleaved. So peptides will generated. So these peptides, these peptides now enters into the ER. ER means rough ER through the tap. Tap means transported associated protein. These are termed it as ABCs. ABCs means ATP binding cassette. So why? Because so here for what is called allowing these peptides, ATP is required. So that's why it is termed it as ABCs. Here it is in this cytosol. This action is in this cytosol. That's why it is termed as cytosolic pathway. So what is the next? Chaperones is comes under the action. Chaperones means here these are regulated the degradation. These acts as a shield to the cell. Why? Because if it is unnecessarily the cell is damaged, it is what is called harmful to the individual. That's why this regulation is achieved by the chaperone. Here, three chaperones is comes participated in this regulation. One is called nexin, another one is called reticulin, third one is stapsin. In this MSC is cytosolic pathway. So class 1 MSC, when it is attaches with called nexin, actually MSC1 contains two structures, alpha chain and also beta 2 microglobulin. So when it is in alpha chain is when it is associated with called nexin, beta 2 microglobulin did not able to bind with the alpha chain. So if it is a peptide enters into the RER, it is a symbol to the connexin. So it unwinds to the alpha chain. Then beta microglobulin will associate to the alpha chain. So when it is associated covalently, it makes covalent bond. It makes covalent bond. So when it is attached to the alpha chain, this beta 2 microglobulin, MSC1 is generated. So now it moves closely to the tap. Tap means transport associated protein. It is achieved by the tapsin. So tapsin is useful to for moving the proximity to the tap. So by this what is used? So these peptides before entering into the lumen, it is enters on the surface of the MHC1. So next chaperone is called reticulin. Actually, it prevents the exit of the from the RER. So now when it is this peptide we attach to the MHC1, it is also allowed for the exiting of the RER. So now this MHC1 will associate with the Golgi complex. From these, it move on the cell, surface of the cell. Now it is a symbol. I am damaged. So kindly kill me. It gives a symbol to the immune system. So then T cells will come into the action. T cells. These T cells contain CD8 receptors. T cells. T cells means cytotoxic T cells. T cells are two types: T helper cells, cytotoxic T cells. So cytotoxic T cells contain CD8 receptor. So it is a MHC1. So these MHC1 is read also POW chemicals on the surface of the cell. So the cell is died. So it is the cytosolic pathway. Students, you thoroughly listen this video. And also, if you have any queries, put it into the WhatsApp. It is a very important one. And also make notes by what is called by writing these slides, by observing the slides and make it on the notes. So it is very useful. Thank you. Tomorrow I will discuss the another topic.